everyone. Today, we are going to discuss molecular coagulation testing. Molecular testing in the coagulation laboratory is available for patients with thrombophilia. Molecular testing is now readily available for gene mutations of factor V and prothrombin for thrombophilia testing. Testing for mutations of the methylene tetrahydrofolate reductase gene is a cause of thrombosis, are also performed to aid in the diagnosis. However, the clinical utility of methylene tetrahydrofolate reductase testing is not clear, and the American College of Medical Genetics and Genomics has recommended that methylene tetrahydrofolate reductase testing should not be routinely performed for the workup of patients with thrombophilia. There are several testing methods available for the clinical laboratory for the more commonly occurring clinically relevant genetic mutation in this table. Common methods are polymerase chain reaction based assay. PCR is accurate for the detection of both point mutation and single nucleotide polymorphism. Factor V laden, prothrombin, and methylene tetrahydrofolate reductase mutations are detectable by this method. A common method to analyze PCR product is restriction fragment length polymorphism analysis. However, RFLP analysis is not a high throughput method, is not suitable for high volume laboratories. Polymerase chain reaction is a technique used to make many copies of a specific segment of DNA. It's very accurate for finding small changes or variations in the DNA, such as point mutations or single nucleotide polymorphisms, which are differences in a single building block of DNA. The mutations related to factor V laden, prothrombin, and methylene tetrahydrofolate reductase can all be detected using PCR. After PCR, one common method to examine the DNA fragment is called restriction fragment length polymorphism analysis. This technique looks at the length of the DNA fragments after they've been cut by certain enzymes. However, while RFLP analysis is effective, it's not ideal for labs that handle a lot of samples because it's not very efficient for processing large numbers of samples quickly. So, while PCR is great for finding genetic mutations, RFLP analysis, though useful, isn't the best choice for labs dealing with a high volume of samples. Other methods for molecular testing in the clinical laboratory that are PCR based or not based that no longer require restriction digestions are also available. A popular non-PCR based method is the invader assay, which uses allele-specific hybridization in a high throughput format. In an effort to obtain rapid and reliable results, several new molecular methods have been developed. Sequence-specific primers, allele-specific oligonucleotides, hybridization, rapid cycle PCR using light cycler instrumentation, and nanochips are now available. In other words, there are various methods for molecular testing in clinical laboratories, some of which are based on PCR, while others are not. Newer methods have been developed that no longer require the step of restriction digestion. One popular non-PCR based method is the invader assay, which uses allele-specific hybridization and can process a large number of samples quickly. Additionally, to achieve fast and accurate results, several other molecular methods have been introduced. This includes sequence-specific primers, allele-specific oligonucleotides hybridization techniques, rapid cycle PCR using specialized instrumentation like the light cycler and the use of nanochips. These methods offer efficient way to analyze genetic material, providing rapid and reliable results for various clinical applications. More recently, high-throughput sequencing has become available for the diagnosis of hereditary bleeding and platelet disorders. The traditional sequencing information used a low-throughput technique called Sanger sequencing. This technique was low, expensive, and had a low sensitivity. HTS allow for the sequencing of multiple DNAs in parallel, resulting in the sequence of millions of DNA molecules at the same time. HTS can sequence an entire exome that can be used to identify variants or mutations in DNA and RNA responsible for different diseases. HTS has become the primary means of identifying genetic variants. The complexity and size of the sequencing data require changes in the diagnostic process such that team composed of clinicians, geneticists, and bioinformaticians are needed. High-throughput sequencing has revolutionized the diagnosis of hereditary bleeding and platelet disorders. Previously, traditional sequencing methods like Sanger sequencing were slow, expensive, and had limited sensitivity. However, HTS enables of this simultaneous sequencing of millions of DNA molecules, allowing for the rapid and comprehensive analysis of genetic material. It can sequence entire exomes, which are all the protein coding regions of genes, helping identify various genetic variants of the mutation responsible for different diseases. As a result, HTS has become the primary method for identifying genetic variants. Nevertheless, the complexity and volume of sequencing data necessitate changes in the diagnostic process, requiring interdisciplinary teams composed of clinicians, geneticists, and bioinformaticians to effectively analyze and interpret the results. Last, the role of molecular diagnostic in thrombophilia and bleeding disorders will continue to grow due to the identification of associated genetic mutation and polymorphism. The challenge for the laboratory is to determine which test to offer and their relevance to patient management. So that's could be all. Thank you.